Hey everyone, it's Mr. Jennings here, and today, in this video, we're going to be talking about, can you alter a gene? And the question is, how do you do it? So, I'm going to zoom out here just so I can get... Okay, so today's topic is probably one of the more interesting ones and the more common ones that we're going to start seeing. This is start getting into more pop culture and general current events. So gene altering is fairly new. Processes go back to life itself, but human application of those processes are fairly recent. We didn't know about DNA really being a thing that we know of today. Well into the 70s. And really, a lot of these tools that we're doing now weren't even in practice until just 10 years ago. So there's a lot of things going on. If you go anywhere on the major news sources, you probably see people talking about this, especially right now with COVID-19, the novel coronavirus. A lot of talks of genetic engineering in terms of how to fight the virus and how to identify possible vaccines and treatments. So let's get into this. We're going to actually a pretty interesting topic. So we're going to talk about how can you alter a gene. And like I said earlier, long story, you can, but how do you do it? So just remember, genes are the DNA base sequence that codes for proteins, and proteins are the ultimate expression of a trait. So the genetic base sequence codes for a protein, which again determines our traits. Now, one of the main things we can do with genes is genetically modify our food. And that's probably the biggest hot topic out of this, like GMOs. Genetically modified foods have been demonized in recent years by various health advocates and environmentalists alike. However, if you look at a history of food cultivation, it's apparent that we've been eating this all along. This is from Science American editor Eric R. Olson. If you can look at that link there, I'll probably try to include it, but it may be behind a paywall. But the idea is, we've been doing GMOs for a very long time. We've been utilizing GMOs for a very long time. So, nothing inherently wrong with them. We'll talk about some of the benefits here in a little bit. So, the reason why this all works is because the genetic code is universal. All living organisms use the exact same DNA information, ATCG. They use the same code book. That means they read everything about it. Basically, they're using the exact same letters and the exact same syntax. Basically, the letters are arranged in a certain way. We speak it and pronounce it the exact same way. While we look so diverse and unique, we all speak the same basic fundamental language. And because we all speak the basic fundamental language, one is common ancestry of life. This points to evolutionary, is a major part of evolutionary theory, which we'll talk about. Two, and the really big thing is, since we all speak the same language, I should be able to take my DNA and put it into another organism, and it should code for the same protein, and vice versa. And it does. So genetic engineering, certain ways we can do this. If we were in the classroom, there's some cool stuff I can do, and I had planned, but the situation kind of doesn't permit. So genetic engineering is the process of altering the genetic code of an organism. For instance, the glowing green protein found in jellyfish, we can actually cut that out and implant it into another organism. So what we did was found the gene for jellyfish that made them glow green, cut out that DNA, and insert that DNA segment into a lab mouse. And when we did that, it glowed in the dark. We even did this to a monkey and did it to a pig. And I could do this in the classroom to bacteria, but I don't have the ability to do that. I was also going to turn it blue. I don't like the glow in the dark one. The glow in the dark one actually doesn't work very well. But yeah, we engineer a lot of organisms. So why do scientists genetically engineer organisms? Mostly medical reasons. It allows for organisms to produce new proteins beneficial to humans. Probably the most famous one is human insulin. If you do not know this, human insulin was produced from cows for the longest time. We used to take bovine insulin. So if you were diabetic, you were at the mercy of the slaughter industry. So what they would do whenever they would slaughter a cow, and they still do this by the way, they would take the spleen of a cow and extract. Now, insulin is a hormone protein that is necessary for regulating body sugar. Blood sugar, sorry, not body sugar. If you, without proper insulin levels maintained, organisms can undergo diabetic shock. Numerous organisms can do this. Dogs, cats, people. Now, when they discovered insulin found in the pancreas, and they determined a way to purify it, it made it a lot easier for diabetics to have some success. The problem is, the demand is so much greater than the supply. You don't get a lot of pancreas from a cattle pancreas. So you're not getting a lot of insulin from a cattle pancreas. So what do? Well, they take the gene for insulin, and they get it out, and they implant it into a bacteria. By implanting it in a bacteria, as you see here, what we do is you identify the gene, say for insulin, you cut it out, you cut the DNA in both organisms. You're going to basically, using a special enzyme, insert a new gene into the organism. The host organism will read the gene as over its own and make that protein. For instance, when we insert genes into organisms to produce insulin, all of a sudden all the cells are going to produce insulin. Now, human cells take too long to grow in a lab. They are notoriously difficult to grow in a lab. They're famous for it. Bacteria cells, on the other hand, will grow if you don't even want them to. So what you do is you take the human part of the DNA, the part of the human DNA that has insulin, this little black segment here. Using a special enzyme, you would cut the bacterial DNA up just a little bit, just big enough to fit that insulin. Insert the insulin gene. Excuse me. And when that bacteria goes to divide, first off, it's going to make all of its offspring have the insulin gene. But every time that bacteria goes to express that gene now, it's going to make insulin. And this is where we come in. As we do this on a large industrial scale, we will then take those bacteria cultures, harvest them. We will purposely kill them off, extract all the insulin out of them, bottle it, purify it, bottle it, ship it out. And there you go. That's how we make insulin in the United States and around the world at a mass scale. We also do this for other proteins like HGH, which is human growth hormone, which is necessary for a lot of disorders, but also can be used as a performance. The same principle goes through here. We also genetically modify chicken eggs a little bit to produce flu vaccine, and it gets into more complicated stuff. Now, the enzymes that we use this for are highly specific things known as restriction enzymes. These are scissor-like enzymes that cut DNA at specific sequences. Basically, they were used by bacteria to cut up DNA for attacking viruses, and there's a variety of them here. These are the three main restriction enzymes that we do. And what they do is they cut to produce what we call a sticky end. Now, the sticky end works because, it allows unpaired bases. So whenever you take the desired trait and move it into place, it has somewhere to attach. And it basically fills in the void. Everything seems nice, and all of a sudden you got yourself a protein that works. Again, GMOs are used for a lot of things besides insulin production. Big thing is we can use it to protect crop from insects. So we put it in corn to help protect it from various insects that may eat on it. The corn will produce a toxin that kills caterpillars, but not humans. We use it for extended growing seasons. Fish berries are basically a type of strawberry that contains anti-freezing gene from a fish known as a flounder. It allows the strawberry to grow longer without worrying about frost as much. And we can also use it to improve quality of food. That's what we see down here in this picture. Golden rice. Golden rice is a staple food crop for billions of people around the world. It is probably the most important single food crop. However, it is nutritionally poor. So what do? They've genetically modified it to produce vitamin A, which is a necessary nutrient that is hard to obtain in most diets in areas where rice is a #1 crop. So by putting vitamin A into the crop, you still produce, and eating the golden rice gives you better nutrition. So, summary of a genetic process here. Step 1, you cut the gene from a human out with an enzyme. Step 2, you cut the bacterial plasmid, which is a type of DNA, with a different enzyme. Step 3, you introduce them to each other. Usually that means the bacteria is going to panic and will get to it, no problem. And the recombinant DNA is then inserted to the bacteria at 4. At 5, allow the bacteria to reproduce. At the end, you're going to produce enough of the desired trait. In this case, humulin is the primary insulin. So you produce insulin. Pretty cool. And then, extract the protein. So genetic engineering is one of the ways we can rapidly produce a chemical, necessary proteins especially, and we find that there's a lot of interesting things we can do. One thing I don't get into right here is a thing called CRISPR, which is a gene editing tool basically that's designed to cut out a section of DNA of a host, insert a new section at roughly the same time of a desired trait, and move on. And that's showing promise to help fight diseases that are embedded in your DNA, such as HIV. So that's pretty cool stuff out there. Alright, that concludes it for now, though. Next up, in the last video for this, we're going to talk about gel electrophoresis. But I hope you're all taking care. See you around. Adios.